This video is going to offer an overview of some of the techniques used in modern biotechnology. Obviously, you can see in the videos, I'll go through these in greater detail, and there are more videos for students in, say, a genetics class. This is meant for more, an introductory biology student or a non-major. So first off, DNA, this is the code for life. And so the better you can study it, the more you can understand life. And there are applications like, identifying diseases in patients, identifying paternity, identifying samples left at a scene of a crime, studying wildlife to find out which ones are more closely related to others. So there are so many applications, as we'll get into. And obviously some of these, I have more humorous examples. But first, you have to make sure that you have enough sample to study. And so the first technique is what's known as polymerase chain reaction, or PCR. See, from say, a blood sample or evidence left at a crime scene or isolated from a fossil or whatever, this might not be enough DNA to test, to say do a DNA fingerprint. And so if you could take this and make more, that would be wonderful. Well, so if you simply heat the double-stranded molecule of DNA, it separates into its two halves. And then each of the halves can then replicate thanks to complementary base pairing. So every at some point, notice that once I separated these two strands, which were once joined, this one and this one, and I let them both replicate, notice each one of them has a blue thymine bound to an adenine, a blue thymine bound to an adenine, etc. And so because each half has specific matches in DNA nucleotides for its complementary bases, then if you separate two strands, let them replicate, now you'll have two copies of the same sequence. Now, because you have to heat this to a high temperature, and because the enzymes, the blue circle in this case, which perform this reaction, would degrade at a high temperature, what one used to have to do is to keep adding new enzyme, which was obviously expensive because each time you heated it, the enzyme became non-functional. The great breakthrough was to use enzymes from bacteria, which live at very high temperatures. And so therefore these enzymes did not then degrade at the high temperatures needed to separate the strands. Now, with this, one could kind of manually then do a number of cycles where you take your sample, you take it from one water bath to another for 90 seconds to 2 minutes to 3 minutes, and then repeat this. And each time you'll get more and more sequence. But there are polymerase chain reaction chambers or cyclers, which will do this on their own. So you can kind of set it up as you're leaving the lab in the afternoon, and it will run overnight and do a number of cycles where each cycle the strands separate, they're allowed to replicate, and now you have double the copies that you had before that cycle began. And this is a way that you can now get enough sample to test. So PCR, polymerase chain reaction, is a way of just amplifying DNA. So now you can do some of the subsequent tests. The next thing that you might want to do with DNA is to cut it so that you could make a series of fragments, which could then be studied. Now, all of us in our digestive systems have enzymes which break down DNA. You do it all the time with chicken DNA, with banana DNA. You break it into pieces and you absorb it for energy. That's not useful because your digestive enzymes do break it up in a different pattern each time. And then they break it up into all of the components of nucleotides, so there's nothing left to study. But bacteria can have enzymes which are defenses against viruses, which only cut specific sequences. They're known as restriction enzymes or restriction endonucleases. And so if you add this enzyme to a sequence of DNA, whether or not cuts are made depends on the sequences. So that top DNA sequence had one of the sequences recognized by the enzyme, so one cut was made. This next sequence has two such sequences that the enzyme recognizes, so two cuts are made, etc. And this is repeatable. So if you were to take the same sample of DNA and a year later, 5 years later, cut it again with the same restriction enzyme, you would get the exact same pattern of cuts. And so these restriction enzymes are needed to take DNA molecules, which are simply too long. Chromosome 1 from one of your cells would be that long. That's too long to study. But if you can break it up into pieces, now that changes everything. And one of the things that you can do with the pieces then is to perform gel electrophoresis, where you're going to get a gel, which is essentially made like Jello. You know, when you make Jello, you add a protein to hot water, you stir it, and then as it cools, it becomes solid in a specific shape. Here we do the same thing. You get a protein oligurose, you heat it, and so then it dissolves in water, but then as it cools, you can put it into a casting tray, which makes this rectangular gel. And if you put it in this little plastic cone, it makes depressions into which your DNA sample can be added. And so the DNA sample is being added then to one side of this gel. And this is the sample which we had cut with the restriction enzymes. And then we've added a couple of dyes just so that we can observe it when it goes through the gel. If we let it run too long, then our sample will go all the way the length of the gel and into the surrounding buffer, and then we lose our sample. So dyes are added so that we can trace the samples. Then we apply electricity because the gel box where we're running this DNA fingerprint now has an electric current going through it. DNA has a negative charge because of the phosphate groups on the DNA nucleotides. And so therefore it will go from the negative pole to the positive pole. And because we've cut it into fragments, we are separating then the fragments according to size, so that the big fragments are moving more slowly. So here we have the DNA which is uncut, and so the big piece doesn't move very far in our allotted time. If our restriction enzyme made one cut, then we have two pieces, and the smaller one moves faster. So that's the small piece, that's the big piece. And then if more and more pieces are made, then we have smaller and smaller fragments which result, and they would travel at different lengths. But now the beauty is here, if these are samples from different animals or different people or whatever, you could then distinguish something. You could say, aha, I can distinguish this source of DNA from this one, or this is a pattern that I recognize. I've seen it before. And so now this DNA, which is, you know, the code of life, you've broken it into these fragments whose patterns now you can study. Why? Well, so for example, you could ask the question, if someone in the family has just been diagnosed with a disease, and if they have, then it might be of interest then to see if anyone else in the family is carrying the mutant alleles. Very often mutations change, you know, the DNA. You could, you know, delete a few bases, add a few bases, you could do a number of things. And so if it changes the size of these fragments, they will travel differently through the gel. And so one could then distinguish between someone who has normal alleles. All right, so this individual here, let's say this is, you know, the mother, she only has two normal alleles of this gene. Let's say that the second sample was the father. He doesn't have any abnormal alleles. He only has mutant alleles. And now if you look at this first child, let's say, they've inherited a normal allele from their mother and a normal allele from their father. Now this is of importance because a lot of conditions, the earlier you can recognize the condition or aspects according to the condition, then the better the outcome for the patient. Right. So if you can identify who's likely to develop cancer, cancer screening is a part of many people's healthcare as they get older. Mammographies or tests for colon cancer, colonoscopies. But if someone was, say, carrying the mutant allele, so obviously I'm being silly, just not to deal with serious medical conditions. But if this was a mutation caused by this mutant allele, clearly not this allele, because the parents who don't have, the parent who doesn't have this condition, then doesn't have that allele. So these would then be normal alleles. This is a mutant one. Then you could see who else in the family might be a carrier of this trait. Because if you could recognize these, then healthcare providers might say, you're now approved to get this screening at an earlier age than most people would. Now, once again, with healthcare, there's always money involved. But just in a perfect scenario, if something reaches an advanced state, like say cancer, well, that's not only very dangerous to the patient, but incredibly expensive to treat. If you can identify those who are at risk, not only is this great for the patient because you could save their life potentially, recognizing something earlier. But then it would also then be cheaper because doing a colonoscopy far cheaper than the expense of removing say a tumor, etc. And so this can be used to diagnose disease. It can be used to recognize individuals who are at risk of disease. It can be used to identify paternity or maternity even. In the old days, court cases would be tied up with paternity cases. They were very difficult to choose. And obviously, being the parent of a child, can involve, great responsibilities. And so therefore, identifying who is the parent or parents of this child and who isn't, that is, a very important thing. But now this is not something which, ties up a great deal of court time and is very complicated. Because if you look at this child's DNA, then some of it would come from the mother and some of it would come from the father. So if you look at the mother's DNA, here's her banding patterns. Notice that the child received this allele from the mother, but not this one. When we make sex cells, the sex cells only have half of our DNA, not all of it. And so, every child doesn't have all of the DNA from their mother, just half. So this child received that from the mother. Notice that this is a piece of DNA the child has, but notice that the mother does not. So this had to come from the father. All right. And so therefore, if you're looking at two men who might be the father and run their DNA, then whoever is the father had to be the individual who provided this band of DNA. This individual could not have, this one did. And as we go further down, the child has this band of DNA that once again came from that parent. And so DNA can be used to solve paternity cases. Very often in criminal, here's obviously a silly example. Not only can you do DNA fingerprinting on children and adults, but also on fetuses. There are procedures which can actually get access to fetal cells. Amniocentesis removes cells from the amniotic fluid. Chorionic villi sampling, or CVS, removes cells from the chorion. These are extra embryonic membranes, not part of the embryo's body, but nevertheless, they came from the embryo. And so if one wanted to know whether a fetus carried, a trait, I'm just being silly here, a werewolf, trait, from a father. If here's, the mutant alleles, here are the normal alleles, and then you could then do a test of the fetus, then one could determine whether the fetus, was going to express a trait or be a carrier for a trait, etc. This is obviously a silly example, but there are a number of disorders which are quite serious. Some, will cause serious disease in an infant when born. Some will ultimately cause an infant or a child's death. Tay-Sachs disease is not curable. And so children born with this disease only have a short number of years to live. And so when parents are planning their families, this might be information that they would like to know in planning. Often at a crime scene, physical evidence is left behind in the form of DNA. This could be blood, this could be semen, this could be skin cells, etc. And then so you have evidence. With PCR, you could make enough of it to test. And then you could then run a DNA fingerprint of this. And then let's say you have several suspects. You could then ask, this pattern of DNA banding, which was seen at the crime scene, does it match any of the individuals which we suspect of the crime? Now, first, a number of things. First, let's say that you say, oh, this matches suspect number 5. Does that prove that suspect number 5 committed a crime? It does not. It proves that this sample, whether it be blood, semen, skin cells, whatever, that this proves that this is this individual's, DNA. So they, didn't necessarily commit a crime, but they do have to explain how come your blood is at the crime scene, how come your skin cells are at the crime scene, and there could be a very good explanation for that. Also, it should be noted that there have been many cases where individuals have been wrongly convicted by a jury of their peers using physical evidence or eyewitness testimony or whatever. And let's say that a jury was, considering whether suspect 4 was guilty of this crime. Well, this evidence clearly shows that this is not suspect 4's DNA. The first time that a DNA fingerprint was used in a criminal case, it was used to exonerate someone, to show that they were not guilty. And a number of individuals who have been convicted, even some convicted and facing the death penalty, were later exonerated and set free when it was shown that the jury made a mistake. So the evidence that they were using, eyewitness testimony or physical evidence, the carpet fibers matched the carpet fibers from the crime scene, or the tire tracks or whatever, that wasn't good enough. And using that evidence, the jury had come to a wrong conclusion. This person who was in prison for committing the crime, that was not their DNA when it was then tested again. So because DNA is so powerful, there are all of these then potential applications. So as we study extinct animals, if they are recently extinct, then one can obtain, with luck, DNA samples from them. So you can get DNA from mammoths. You can get DNA from insects trapped in amber. There have even, been DNA isolated from fossil bone of, say, Neanderthal humans, or another species of human which lived in Asia, Homo. I'm sorry. the Denisovan humans, which had lived in Central Asia. And, one can now study how they are related to modern organisms, which is fascinating. One can study modern organisms and see, which birds belong in the same family because of their close relations, or, how these insects are related to other insects by comparing genetic similarities. One can apply this to wildlife cases. So if someone says, no, I did not get these animals from the wild. I raised them in my basement. They're all siblings. Well, you can do genetic tests to see if they are all siblings or if they were poached from the wild. If you're, at a market and you buy meat and someone says, oh, this is chicken, you can test to see if it's chicken or an endangered species. If you have an endangered species in a zoo and would like to mate that individual with another member of a species that would get the healthiest offspring, you could do DNA fingerprints of, say, other zoo animals to see which one would be most likely to result in the healthiest offspring. And so DNA fingerprints, since DNA is the code for life, now that we have the ability to study it, it just gives us this, this wonderful ability to better understand life. Now I'm going to go into another set of categories. Once again, this is meant to just kind of be an introduction and by no means a comprehensive introduction into this powerful tool. And while I'm waiting for this to load, it is a tool. So for example, here is an application of DNA fingerprinting. We could take everyone who's, say, convicted of a certain crime, or we could take, every infant at birth and take a sample of their DNA and then put it in a DNA database. And this would have a number of potential applications. So, for example, if ever in the future, say, blood was found at a crime scene, you would now be able to identify whose blood it was because you have a database of, say, everyone. Or you would be likely to find, someone who has already been convicted of a crime because you know what their DNA looks like. Otherwise, you would say, this is someone's blood who has a DNA banding pattern that looks like this. I just don't know who that is. So that's an application of the tool, but is it a correct application of the tool? This is an example I use in my classes. A hammer. What is the morality of a hammer? It doesn't have morality. If you hit a nail on a roof, that's good. If you hit someone in the head, that's bad. So society then makes rules. Hammers are tools. They are amoral. This use of the hammer is permitted. This use of the hammer is prohibited. And the same thing here as we look at these aspects of biotechnology. These are tools. How should these tools be used? Well, that's what society decides. And so, we debate, we elect officials, we have hearings, there are, publications, et cetera. And so we then debate, do we want this tool used for that or not? And so once again, just because there is a tool doesn't mean it has to be used for that. And so if you feel strongly about something, then you should get involved. Another just quick application is we can then probe this, so you can get probes which stick to DNA or RNA or proteins when you run gels, because not only can you do a DNA fingerprint, you can do a gel with RNA, you can do a gel with protein, and then probe for various things, and answer great questions like, which proteins which protein is the cancer cell expressing inappropriately. So there are a number of different ways that you can probe samples. Sorry for that glitch. Another big application is if DNA is the code for biological information, one can actually move biological information from one organism to another. So for example, let's say we cut this bacterial chromosome with one of those restriction enzymes. Well, we cut it and we make specific, sticky ends, they're called. So this strand is looking to bind to a pink adenine, a pink adenine, a blue thymine, a blue thymine. If you cut a human gene with the same restriction endonuclease, you can then insert that here, because DNA is just DNA. It doesn't care. It's just looking for adenines and thymines to bind to. This bacterial chromosome doesn't care that the complementary nucleotides came from a human gene. Once you've done this, you now have a human gene inside bacteria. Who on earth would want to do that? Well, there are lots of human molecules which are valuable, like insulin, which is needed for diabetics, or a number of molecules like interleukins or interferons, which could be used for cancer patients, or alpha-1 antitrypsin. So there's all of these molecules that you might want to treat a patient with. Now, getting human molecules can be difficult. So, for example, if in a couple decades ago, if you wanted to treat a child who was never going to reach their full potential in height because they had a genetic mutation and they couldn't make the hormone, growth hormone, you could give them growth hormone from other individuals, but the treatment might involve getting 50 cadavers a year and extracting the pituitary glands. Not only very expensive, but this meant that many children could not be treated for their disease. There wasn't enough human insulin to go around. So very often diabetics, decades ago, would be getting insulin from slaughterhouses, from other animals, but because it was a hormone from a different animal, not a human, there was a problem. But now, if there's a molecule that you need, these bacteria now make it. They now make human growth hormone or human insulin. And so you can have a lab where you have a big vat of bacteria producing something that humans need in the hospital. And so you can actually take a gene and add it to a bacteria. And there are a number of ways to do that. You could do the same to plants. And so you could say, I would like this, plant, whether it grows tomatoes or, whatever, to have a new trait. I would like it to say be herbicide resistant, because there are so many weeds in this area that, cuts how much profit I can make, but also I have to play all of this pesticide. Right, I screwed this up. So I'm sorry, in the video, I apologize, I made a mistake in the animation. So the herbicide resistant strain would be likely to survive when you applied a herbicide. So you could apply the herbicide that killed the weeds, but not the crop plant. In addition, you could add nutrients to plants to make them healthier. You could say, oh, let's add vitamin A, which is needed in this area where lots of people suffer from vitamin A deficiencies. Let's make this plant better. The previous tomatoes, they would not ripen on the vine. And so they were picked when they were green, and they artificially ripened, or they didn't taste as sweet. If you just remove an enzyme from tomatoes so that they rot more slowly, then they can ripen on the vine. And it's actually, in that sense, more natural. And so there can be all of these things that one can add to plants and change them. So not everything has to be eaten. You could engineer plants to be a biofuel. If you want to release less pesticide, well, if plants can release pesticide from their roots, say when attacked by an insect, now this would prevent you from, say, spraying pesticide on a field of crops where it would get into aquatic ecosystems and food chains, etc. And so it's the same principle. So if you simply cut the DNA of the plant, and then you have this new trait, we'd like to make this plant different in some way, here's the gene which can then be added. Now, not only can this be done to plants, the same principle as what I had just explained, but this could be done to animals as well. You could say, I would like this animal better if they then had this trait. So you could take animal DNA and change it, all right, through the same principles. Now, this could be an animal that you intend to eat, so that you have then changed its DNA so that, perhaps it is meatier and has less fat. Perhaps it grows to an adult size more quickly. But there are other applications as well. So in addition to changing these animals with the idea that this will make them different when we eat them or prepare them to be eaten, these mammals also produce milk. So one of the things that one could do with these animals is genetically engineer them so that they're producing something in their milk that you would like, say, for the hospital. Say these human patients which are going to be treated, they need something. Well, if these animals could be genetically modified, so they're making this anti-cancer, cytokine hormone in their milk, then in your field, you can have a field, a field full of a herd of these animals, and they're not, being harvested for meat. They're just being milked every day, and a product of what they produce can be sold then to the hospital. Now, as I had said, these are tools. Let's say that you have a reaction to something that I've just said, where you say, I feel that, something that you just said is inappropriate, where I don't think, this, should be done. I wouldn't eat that or whatever. I wouldn't eat engineered plants, or whatever. Well, a couple of quick comments. One, that's fine. Once again, our society debates the correct application of a tool. When you say we can use the tool for this purpose, but not for that purpose. And so if you have an opinion on the matter, well, then good for you. And if you would like to get socially active and say, I don't want my supermarket to carry this, or I would like there to be a law against this or whatever, you're free to do so. So there is no application of this that has to be. I do sometimes, throw a couple of just examples. Once again, not trying to convince anyone of my opinions, but just for discussion in my classes. First off, some people will say, oh, this is, quote, playing God. I don't think we should mess with tomatoes or bananas or pigs or chickens or whatever. One comment that I sometimes make is, well, go to a supermarket and tell me what you find that's natural. All right. And you'll find that there's very little. There are no such things as wild chickens. All right. Humans have invented the chicken. Now, it took a long time, but breeders specifically said, we are going to select these chicken ancestors which have these traits that we like, and we're going to make them together so the next generation is a little bit different than the one before, and the generation after that is a little different from the one before. Wheat is a human invention. All right, there was no wild wheat. People said, let's cross this strain from this strain, and these hybrids now which have these extra chromosomes. A lot of the seedless plants that you find in the supermarket are triploid. They have abnormal numbers of chromosomes. That's why they don't make seeds. And so if your idea is I'm only going to eat natural things, well, to be honest, that that's difficult to apply in a supermarket. Humans have been modifying apples and cows and pigs and racehorses and dogs for thousands and tens of thousands of years. I would like this fruit better if it was sweeter. I would like it if it grew in this soil. I would like this horse if it was faster. I would like this dog if it was better with kids or better at herding sheep. And so humans have modified nature for thousands of years. This is just a technique to do it in a more modern sense, which is faster. Now, you can disagree with it. But nevertheless, it's not fundamentally, different. Humans have always changed the life around them. This is just another technique. And I said, I like this application, it's nice being myself environmentalist, but you have analysts say, plants because the site is being used in the environment, there's now less in food chains, etc. One of the so talking about herbicide resistance here, one of the issues which comes up is we certainly live in a big planet. And whatever your feelings on the matter is, you're free to say, I'm going to shop here where they, do this or don't do this. But the world is a big place. And as we have more and more people on the planet, 18 million people on the planet, and so almost 7 billion. Well, how be honestly, not really good. As I saw, tens of thousands of people die of a disease. Maybe you feel I do not want to herbicide resistant so that now I can grow them in this area where this weed was a problem without spraying pesticide. I do not want to eat dry lands corn, which can now be grown in areas of the Midwest that previously could not sustain corn because there wasn't enough water. So here you see how green some of these areas are. That's either one of two things. One, because it's being irrigated from the Ogallala aquifer, whose level keeps going down. Or two, because it's genetically engineered corn in some of these examples, which has been engineered to grow using less water. So you can maybe say, I don't want to eat that, but not everyone has that luxury, all right? And so this is a tool, and even if you of the mind that I don't know where my protein comes today, I don't necessarily have to be bad. Just again, samples applications, and once again, you can certainly make up your mind as to which of these you favor or not, etc. Now, not only can one add new genes to bacteria, to plants, to animals, one can also add new genes to humans. Now, certainly, that there are science fiction stories, where this leads to horrible ends. But there are lots of people who suffer from a genetic disease. They are lacking the ability to make a gene that most people do. And as a result, their life might include more suffering, or their life might be considerably shorter than it otherwise would be. But if you could add a new gene, it might mean that they are now cured, all right, that they no longer have a genetic disease. So this is a successful treatment. Or more commonly, it might not be a cure, but it might make the individual better. So for example, there are a number of cases where an individual who has a disease would have received this treatment, all right, and that would have improved their disease, say, up to a certain level. There's a gene therapy, which you'd hope would cure the disease, but it doesn't. But the patient is better. So if you have the gene therapy alone or the regular treatment alone, the gene therapy and the traditional therapy are better, with whether it be longevity or needing fewer transfusions or, whatever the disease is. So even if gene therapy isn't a cure, when used in conjunction with other treatments, then it leads to better health outcomes. Now, new genes can be added in a number of ways. When I teach genetics, there's a very simple way that we can do, high schools often do. So this doesn't have to be complicated. But in recent years, there is a new a technique known as CRISPR. So this is just one more way that DNA can be edited. And I have a couple of videos which go through the procedure of CRISPR, making it a little easier to edit the DNA of an organism. And so it was really only about 60, 70, years ago that we really didn't understand the genetic code. We didn't understand how DNA worked. A century ago, we didn't even know what DNA was. And so here we are to the point where we understand DNA, where we can study it, we can use it to understand ourselves and the world around us. We can even edit it. And rather than taking generations and generations to say, I'm going to try to develop a breed of dog that does this, or develop a breed of crop plant which is bigger or sweeter or whatever, it can be now done in a much shorter period of time. So, this is, a wonderful, some countries, we approve of this. This just